So this topic talks about the systematics of fungi. For content, this topic can enumerate how living organisms are described. It can explain the principles of classifications and diversity of life forms. It enumerates the economic importance of fungi, and also it describes the diversity of form, functions, and adaptations of fungi. Enumerate the molecular methods used in systematics. Explain how archaea and bacteria are being classified or organized. Appreciate the diversity of living organisms and its contribution to the ecosystem. Now, first question, what is biodiversity? So, when we say biodiversity, it is all the different kinds of life you find in one area. The variety of animals, plants, fungi, and even microorganisms like bacteria that make up our natural world. Each of these species and organisms work together in ecosystem like intricate web to maintain balance and support life. Systematics and biodiversity aims to document the diversity of all groups in living organisms through systematic papers that have broad context and that address the topical issues relating to biological collections, museum science, and principles and practice of systematics and related fields including phylogenetics. The formal biological definitions of species is a group of actual or potentially interbreeding organisms. This means that members of the same species are similar enough to each other to produce fertile offspring together. By these definitions of species, all human beings alive today belong to one species or Homo sapiens. All humans can potentially interbreed with each other but not with the members of any other species. In the real world, it isn't always possible to make the observations needed to determine whether different organisms can be interbreed. For one thing, many species reproduce asexually, so individuals never interbreed even with members of their own species. When studying extinct species represented by fossils, it is usually impossible to know whether different organisms could interbreed. Therefore, in practice, many biologists and virtually all paleontologists generally define species on the basis of morphology rather than morphology. Refers to the form of structures of organisms. Now, for classification purposes, it generally refers to relative obvious physical traits. Typically, the more similar to one another the different organisms appear, the greater the chance that they will be. Now, when we see morphology, it is the study of size, shape, and structures of animals, plants, and microorganisms, or the relationships of their constituent parts. Now, when we say phylogeny, it is the history of evolutions of science or group, especially the reference to lines of descent and relationships among broad groups of organisms. Now, let's go back here to now the diversity of life forms. These These are also often referred as components of systematics. These principles or components are integral in constructing a systems, a classifications which give the best possible interrelations of the taxa, the components like identification, describe, and nomenclature enables a better management of information to be utilized by different people working on different aspects of various taxa. It can be done by either sending the specimen to a subject expert in the relevant field or by visiting herbarium and comparing the specimen with already identified specimens that are present in the herbarium. By use of already published literature of an area having descriptions of similar kinds of plants such as floras, revisions, monographs, and manuals. First, the plant is identified with the help of case, and later the detailed descriptions of the plant is compared with the unknown specimens. Use of virtual herbarium, where one can go through various images which have been already uploaded by experts or amateurs, and the members of a particular group, helps in the identifications of unknown plants. Now for nomenclature. The nomenclature is the determinations of correct name of a taxon is known as the nomenclature. Now, for naming a different groups of living organisms, there are different rules which are followed. For nomenclature of the plants, the rules for International Code of Nomenclature or ICN are already followed. Now, the advantage of having this code is that the only single correct name is available for a particular taxon. Similarly, the nomenclature of cultivated plants is governed by International Code of Nomenclature for Cultivated Plants, ICNCP. Names of bacteria by International Code for the Nomenclature of Bacteria, also called the Bacteriological Code or BC. Now for viruses, there are separate code for a viruses called the International Code of Virus Classification and Nomenclature or ICVCN. Now recently in the year 2007, a rank list of phyllocode has also been developed based on the concepts of phylogenetic systematics. On the basis of recognitions of monophyletic group, only species are recognized. Now for phylogeny. So when we say phylogeny, it deals with the evolutionary descent of taxonomic group and studies of ancestral relationships and lineage. These relationships are often depicted through a diagram known as the phylogram. A phylogram is a branching of a diagram based on the degree of apomorphy. In the descendants, the longest branch represents the most derived group. Geological timescale necessarily may not be there. Monophyletic groups, including all the descendants of a common ancestors, are recognized and from entities in the classification system. Paraphyletic groups wherein some descendants of a common ancestors are left out, are reunited. Now, the phyletic information may often help in determining a phylogenetic relationships. Now, for classification. The classification, it is an arrangement where organisms are grouped on the basis of their similarities. These groups are again assembled into a more inclusive groups until all the organisms have been assembled into a single most inclusive group. In this process, the groups are assigned to a fixed hierarchy or a category such as the species, genus, family, order, class, and divisions. The final arrangement does conclude a classification. Now, there are different types of classification that have been proposed based upon the different characters. That some important one are: first, the artificial classification. Second, the natural classification. Third, the phenetic classification. And then lastly, the phylogenetic classification. First, we have the artificial classification. So the artificial classification, these systems of classifications is also commonly known as the sexual systems of the plant. Classification here are artificial characters were taken from the classifications of plants into various groups. This artificial classification, this system was proposed by Linnaeus, who used the numbers of stamen group of the plants. Second is the natural classification. This system uses overall similarity in grouping taxa. This concept has started by M. Adanson and was later comprehensively used by Betham and Hooker. In this system, mainly morphological features were used to delimit similarities between the plants. Third is the phenetic classification, a system of classification that does not try to reflect evolutionary relationships. Instead, it is based on the physical similarities among organisms or the phenotype. Organisms are placed in the sum category category because they look alike. Smith and local and Sokal strongly advocate these systems of classifications but did not find much favor by others. And then lastly is the phylogenetic classification. The phylogenetic classification are based upon the evolutionary descent of a group of organisms. Diversity of form, functions, and adaptations in fungi. Fungi contain a distinctive adaptations. Adaptations is that the ability to secrete exoenzymes that area unit accountable for digesting organic materials outside of the plant body. Once the digestion takes place, the plant life can then ingest the fabric and utilize it consequently. The fungi contain a distinctive Now for hyphae and mycelium, the mycelium and the hyphae are both important parts of fungi, commonly known as the mushrooms. The two terms refers to the building blocks of fungi. Hyphae, or the plural form singular from hypha, is the name of the long individual pieces that comprise a mycelium. So, the hyphae are often described as strings and thread-like filaments. The hypha is basic body of a comprised of one or more cells surrounded by a tubular cell wall. Now, on the other hand of mycelium, the plural form mycelia, is the vegetative part of the fungus in relation to the hyphae. It is the network of collections or bundles of hyphae in one single place. Unlike the hyphae, the mycelia can be visible to the naked eye in the form of mold on various. Now we have the colonizations of substrates. The term substrates gets used in two senses. In the first place, it can refer to the substance that the fungus uses as food. The mycelium of the fungus grows through this kind of substrate, secreting enzymes that dissolve part of the substrates, the absorbing the nutrition that results. The growing through leads us the second sense, which is whether sort of substance, the fruiting body, or other visible token of the fungus. appears on usually these two senses refers to the some substance but not always. Now let's talk about the sexual reproductions and the asexual reproductions. So the sexual reproductions in the fungi consist of three sequential stages. First, the plasmogamy, karyogamy, and meiosis. The diploid chromosomes are pulled apart into two daughter cells, each containing a single set of chromosomes or the haploid state. Second, the plasmogamy, the fusions of two protoplast. Protoplast is the contents of the two cells. Brings together two compatible haploid nuclei. At this point, the two nuclear types are present in the same cells, but the nuclei have not yet fused. Then third, the karyogamy. It is the results in the fusions of these haploid nuclei and the formations of a diploid nucleus. Nucleus contain two sets of chromosomes from chromosomes, one from each parent. The cell formed by karyogamy is called the zygote. In most fungi, the zygote is the only cells in the entire life cycle that is diploid. The eukaryotic state that results from plasmogamy is often a prominent condition in the fungi and may be prolonged over several generations. In the lower fungi, karyogamy usually follows plasmogamy almost immediately. In the more involved fungi, however, ang karyogamy is separated from plasmogamy. Once karyogamy has occurred, meiosis, the cell divisions that reduce a chromosome's numbers on one set per cell. So the meiosis generally follows and restores the haploid phase. The haploid nuclei that results from meiosis are generally incorporated in spores called meiospores. Now for the asexual reproduction. So fungi reproduce asexually by fragmentation, budding, or producing spores. Fragments of hyphae can grow new colonies. Mycelia fragmentation occur when the fungal mycelium separates into pieces, with each component growing into a separate mycelium. Pomatic cells in the yeast from buds. During bud, a bulge forms on the side of the cell. The nuclei divide and the bud ultimately detaches itself from the cells. And then the most common mode of sexual reproductions is through the formations of asexual spores, which are produced by one parent and or genetically identical to the parent. The spore allow fungi to expand their distributions and colonize new environment. Now, what is spore dispersal? So, the fungi produce their spores in usually liver brown slime, which is on top of a colorful part of the fruit body. When the spores are mature and exposed to the external environment, the odor of the spore will attract the flies that will eat up the slime and spore, thereby dispersing the fungus. Fungal spores and pollens can be dispersed into a number of ways. by animals and insects, by water, by wind, or by rain. Now for the nutritions. Fungi get the nutritions by absorbing organic compounds from the environment. Fungi are heterotrophic. They rely solely on a carbon obtained from the other organisms of their metabolisms. The nutritions Fungi obtain nutrients in three different ways. First, they decompose dead organic matter. Saprotroph is an organism that obtains its nutrients from non-living organic matter, usually dead and decaying plants or animals' matter, by absorbing soluble organic compounds. The saprotrophic fungi plays very important roles as recyclers in ecosystems, energy flows, and biogeochemical cycles. Second, they feed on a living host. As a parasites, fungi live in or on other organisms and get their nutrients from their host. Parasitic fungi use enzymes to break down living tissues, which may causes illness in the host. Diseases causing from fungi are parasitic. And then lastly is they live which mutualistically with other organisms. Mutualistic fungi live harmlessly with other living organisms. Recall that mutualism is an interactions between individuals of two different species in which both individuals benefits. The adaptation, fungi contain a distinctive adaptation. Adaptation is the ability to secrete exoenzymes, that area unit accountable for digesting organic matter outside of the plant body. So for adaptation, once digestion takes place, the plant's life can then ingest the fabric and utilize it consequently. Fungi have adapted over the years in response to their environment. One way in which they have adapted is by increasing their surface area of their gills. This beneficial to the organisms because it is able to reproduce more spores, which can lead to more of them being dispersed. This dispersal can lead to a better survival of the fungi. Another way in which they have adapted over the time is by increasing the thickness of the spores walls that are already dispersed. Now, for having more protections for the spores allows them to be able to survive better once they land on the new ground In times of drought, this can be essential in the survival of the new organisms. Environ The economic relevance of fungi. Fungi are an important organisms in human life. They play an important role in medicine by yielding antibiotics, while in agriculture by maintaining soil fertility, are consumed as a food and forms the basis of many industries. Now let us have a look at some of the fields where fungi are really important. Now here, the importance in human life. These microbes along with bacteria bring about recycling of matter by decomposing dead matter of the plants and excreta of animals in the soil. Hence, their use of enriches the soil to make it fertile. The absence of activities of fungi can have an adverse effect on this ongoing process by continuous assembly of fungi are very important to humans at many levels. They are an important part of the nutrient cycles in the ecosystem. They also act as the pesticides. Biological insecticides, fungi are animal pathogens. Thus, they help in controlling the populations of pests. These fungi do not infect plants and animals. They attack specifically to some insects. The fungus Beauveria bassiana is a pesticide that is being tested to control the spread of emerald ash borer. Now, the importance in medicine. Metabolites of fungi are of a great commercial importance. Antibiotics are the substance produced by fungi useful for the treatments of diseases caused by pathogens. Antibiotics produced by actinomycetes and molds inhibits the growth of other microbes. Apart from curing diseases, antibiotics are also used to feed to animals for speedy growth and to improve meat quality. Antibiotics are used to preserve freshly produced meat for longer duration. Penicillin is a widely used in antibiotics, lethal for the survival of microbes. The reason it is extensively used is since it has no effect on human cells but kills gram-positive bacteria. The streptomycin, another antibiotic, is one of the great medicine value. It is more powerful than penicillin as it destroys gram-negative entities. The fungi plant dynamic is essential in productivity of crops. Fungal activity in farmlands contributes to the growth of the plants by about 70%. Fungi are important in the process of humus formation as it brings about the generations of the plants and animal matter. They are successively used in biological controls of pests. Plants pests are used as insecticides to control activities of insects. For example, the Impausa sepulchralis, Cordyceps melonhae, it is use of fungal pesticides can reduce environmental hazard by a great extent. Fungi are also used in agricultural research. Some species of fungi are used in the detections of certain elements such as the copper and arsenic in soil and in the productions of enzymes. For instance, biological and genetic research on fungi named Neurospora lead to the one gene, one enzymes hypothesis. The fungi live in a symbiotic relationships with the plant roots known as the mycorrhiza. These are essential to enhance the productivity of farmland to 80 to 90%. Of trees could not survive without the fungal partner of the root system. So that is the importance in agriculture. Now for the importance in food industry. Some fungi are used in food processing while some are directly consumed. For example, the mushrooms, which are rich in proteins and minerals, are low in fat. Fungi constitute the basis in the baking and brewing industry. They bring about the fermentations of sugar by enzyme called zymase, producing alcohol which is used to make wine. The carbon dioxide or the byproduct in the process is used as a dry ice and also in the baking industry to make the dough, rising and lighting of dough. Saccharomyces cerevisiae is an important ingredient in bread and this unstaple food of humans for several years. It is known as the baker yeast. Evolutionary and molecular taxonomy. Molecular taxonomy is a branch of the science that deals with the presence of chemical inside our body. It is method of identifying and distinguishing the specimens in a fast and extract manner which is based on the interspecific values. In phylogenetic analysis, branching diagrams are made to represent the evolutionary history of relationship between different species, organisms, or characters of an organisms, or the genes, proteins, organs, or etc. So the characteristics of organisms that are developed from a common ancestors. The diagram is known as phylogenetic trees. Phylogenetic analysis is important for gathering information on biological diversity, genetic classifications, as well as learning developmental events that occur during the evolution. With advancements in genetic sequencing techniques, phylogenetic analysis how involve the sequence of genes to understand the evolutionary relationships among species. DNA being the hereditary material can how be sequenced easily, rapidly, and cost-effectively. And the data obtained from genetic sequencing is very informative and specific. Also, morphological estimates can be used to evolutionary developments, especially in cases where genetic material is not available or the fossils. Systematics of archaea and bacteria. The late 19th century was the beginning of bacteria taxonomy. And bacteria were classified on the basis of phenotypic markers. The distinctions of prokaryotes and eukaryotes are introduced in the 1960s. Numerical taxonomy improved phenotypic identification but provide little information on the phenetic relationships of the prokaryotes. Later on, the chemotaxonomic and genotypic methods were widely used for a more satisfactory classification. Archaea were first classified as a separate group of prokaryotes in the year of 1977. The approaches to the classifications of prokaryotes or the prokaryotic. Now, even though the prokaryotes are tiny organisms, they differ greatly in where they live, what they use for the food, and their DNA sequences. Past scientists classified prokaryotes into two groups called Eubacteria and Archaebacteria. The kingdom of Eubacteria was made up of the everyday bacteria or the everyday bacteria, such as the ones that make milk sour, decompose dead organic matter, and on occasions make people sick. The kingdom Archaebacteria was made up of a strange group of organisms that scientists found extremely hot, acidic, or salty environment. In the late 1970s, the two American scientists Carl Woese and George Fox produced the Archaebacteria were not just a weird bacteria, but instead were a distinct group of organisms that are very different bacteria. Now, how many prokaryotic species are in nature? Are there in nature? The global diversity of bacteria and archaea, or the prokaryotes, the most ancient and most widespread forms of life in Earth, is subject to high uncertainty. To estimate the global diversity of prokaryotes, a study was to analyze a large number of ribosomal RNA ring sequence found in all prokaryotes and commonly used to catalog prokaryotic diversity. Using several statistical approaches and through a comparison which exists with existing databases and previous independent surveys, we estimate they estimate that there exists globally between 0.8 and 1.6 million prokaryotic. When restricting our analysis to the Americans while controlling for the numbers of studies obtained similar estimates as for a global data set, suggesting that most OTUS are not restricted to a single continent but are instead globally distributed. The estimates contain the extent of a commonly hypothesized but poorly quantified rare prokaryotic biosphere and refute recent predictions that there exist trillions of prokaryotic OTUS. The findings also indicate that contrary to a common speculations, extinctions may strongly influence global prokaryotic diversity. The second topic is about the algae biophyte systematics. For content, this topic can enumerate and describe the phylum of the kingdom Fungi, explain and enumerate approaches using classifying plants, describe how plants are being classified in early years, enumerate the 5 groups of the plant kingdom. It also explained how algae are being classified, appreciate the importance of plants in the society. Now, plant systematics is a science that includes and encompasses traditional taxonomy. However, its primary goal is to reconstruct the evolutionary history of the plant life. It divides plants into taxonomic groups using morphological, anatomical, embryological, chromosomal, and chemical data. However, the science differs from a straight taxonomy in that it expects the plants to evolve and documents the evolutions. Determining phylogeny, the evolution's history of a particular group, is the primary goal of the systematics. The systematics and fungi. Fungi are usually classified in 4 divisions. The first division is the Chytrodimycota or the chytrids. zygomycota or the bread molds, ascomycota or the yeast and sac fungi, and the basidiomycota or the clove fungi. This division is I already discussed from the past discussions. So the placements into a division is based on the way in which the fungus 38 reproduces asexually. Unlike other fungi, the lichens are not a single organisms but rather a symbiotic association between a fungus and an algae. The shape and internal structure of the sporangia, which produce the spores, are most useful character for identifying these various major groups. There are also two conventional groups which are not recognized as a formal taxonomic group. They are the polyphyletic. These are the Deuteriomycota or the fungi imperfecti, and the lichens, Deuteriomycota includes all fungi which have lost the ability to reproduce sexually. As a result, it is not known for a certain into which group they should be placed, and thus the Deuteriomycota becomes a convenient place to adapt them. The systematics and fungi, the fungal members of the lichens, is usually an ascomycetes or the basidiomycetes, and the alga is usually a cyanobacterium or the chlorophyte or the green algae. Often the fungal partner is unable to grow without the algal symbiont, making it difficult to classify these organisms. They will be treated as a separate group, but it should be realized that they are neither single organisms nor a monophyletic group. It should also be noted that some organisms carry the name of molds or fungus but are not classified in the kingdom of the fungi. These include the slime molds and water molds or the Omycota. The slime molds are now known to be a mixtures of 3 or 4 unrelated groups, and the Omycetes are now classified in the Chromista with the atoms are brown algae. For the phylum Chytridiomycota or the chytrids is one of the 5 true phyla of fungi. There is only one class in the phylum Chytridiomycota, the Chytridiomycetes. The chytrids are the simplest and the most primitive Eumycota, or the true fungi. The evolutionary record shows that the first recognizable chytrids appeared during the late of Precambrian period, more than 500 million years ago. Like all fungi, chytrids have chitin in their cell wall, but one group of chytrids has both cellulose and chitin in the cell wall. Most chytrids are unicellular, a few form of multicellular organisms and hyphae, which have no sept between cells. They reproduce both sexually and asexually. The sexual spores are called diploid zoospores. Their gametes are the only fungal cells known to have a flagellum. For the phylum Zygomycota, the zygomycetes are a relative small group in the fungi kingdom and belong to the phylum Zygomycota. They include the familiar bread mold, Xyropos stolonifer, which rapidly propagates on the surface of the bread, fruits, and vegetables. They are most terrestrial in habitat, living in soil or the plants and animals. Most species are saprobous, meaning they live off decaying organic materials. Some are parasites of the plants, insects, and small animals, while they are asymbiotic relationships with the plants. Zygomycetes plays a considerable commercial role. The metabolic products of other species of rhizopus are intermediates in the synthesis of semisynthetic steroid hormones. Now for the phylum Ascomycota. Ascomycota are separate fungi with the filaments partitioned by cellular cross walls called septa. Ascomycetes produce sexual spores called ascospores, formed in sac-like structures called asci, and also small asexual spores called conidia. Some species of Ascomyta are usually and do not form asci as ascospores. Condyneal or mytosporic fungi. The mytosporic fungi are a group of a fungi that consists of sexual states of the most Ascomycetes and some Basidiomycetes. The mytosporic fungi undergo mitotic divisions to produce microscopic asexual structures called condia, which are of the various colors, size, shape, and septations. Next is the phylum Basidiomycota. The phylum Basidiomycota is a group of fungi with many different species. It includes mushrooms, puffballs, stinkhorns, jelly fungi, and yeast. Basidiomycota are the filamentous fungi that are made of hyphae except for yeast. They reproduce sexually by formations of specialized club-shaped cells called basidia. Ang basidia, it is the normal produce for a haploid spore called basidiospores. The systematics of botany. Plant systematics is an ancient science based firmly in the empirical tradition. It has its roots in the study of medicinal plants and agriculture. The formulations of the modern classifications involves the accumulations of taxonomic data via observations and experimentations. From these data, generalities on evolutionary relationships are induced and classifications or classificatory hypotheses formulated. Classifications change when new data falsify the existing ones. Classification may change, but names for the most plant remain constant. Within any given classification, each taxon can bear only one specifically valid name. DNA sequence data challenge most of our major classificatory concepts. Next is the history and development of the plant systematics. Plant systematics is the science that encompasses traditional taxonomy. However, its primary goal is to reconstruct the evolutionary history of plant life. It divides plants into taxonomic groups using morphological, anatomical, embryological, chromosomal, and chemical data. However, the science differs from a straight taxonomy in that it expects the plants to evolve and documents that evolution, determining phylogeny or the evolutionary history of a particular group is the primary goal of the systematics. Now, for studying plant systematics, the International Associations for Plant Taxonomy, located in Bratislava, Slovakia, seeks to promote botanical systematics and its significance to understanding and value the biodiversity. They publish a biomonthly journal devoted to a systematic plants biology. In the USA, the University of Chicago Botanic Garden has a plant systematics laboratory. They seek to put together accurate information about the plant species so as to describe of them for a research or restoration. They also keep preserved plants in the house and date when they are collected in case that is the last time the species is over collected. The plant diversity. Kingdom Plantae includes all the plants. They are eukaryotic, multicellular, and autotrophic organisms. The plant cells contains a rigid cell wall. Plants have chloroplast, chlorophyll pigment, which required for a photosynthesis. The plant kingdom has the following characteristics features. First, they have a non-motile. They make their own food, hence are called autotrophs. They reproduce asexually by vegetative propagations or sexually. These are multicellular eukaryotes. The plant cell contains the outer cell wall and the large central vacuole. Next, the plants contain photosynthetic pigments called chlorophyll present in the plastids. And then lastly, they have different organelles for anchorage, reproductions, support, and photosynthesis. Now, the classifications of the kingdom Plantae. First, the plant body, presence or absence of a cell-differentiated plant's body, the root, the stem, and the leaves. Second, the vascular system. The vascular system, presence or absence of a vascular systems for a transportations of water and other substances, which is the phloem and xylem. And then third, the seed formation, presence or absence of the flowers and seeds, and it is the seeds are naked or enclosed in a fruit. We have the Thallophyta. Thallophyta includes plants with primitive and simple body structure. The plant body is thallus. They may be filamentous, colonial, branch, or unbranched. For example, include the green algae, red algae, and brown algae. Next is the Bryophyta. The Bryophytes do not have a vascular tissues. The plant body has root-like, stem-like, and leaf-like structure. Bryophytes are terrestrial plants but known as the amphibians of the plant kingdom as they require water for a sexual reproduction. They are represent in most and shady places. Sorry. So the bryophyta includes mosses, hornworts, and liverworts. Next is the Pteridophyta. The Pteridophytes have a well-differentiated plants body into a root, stem, and leaves. They have a vascular system for a conditions of water and other substances. Next is the gymnosperms. The gymnosperms have a cell-differentiated plant body and vascular tissue. They have a bare naked seeds. These seeds are not enclosed within the fruit. Next is the angiosperm. Angiosperm are the seed-bearing vascular plants. With a well-differentiated plant body, the seed of the angiosperm are enclosed within the fruits. Angiosperm are further divided into a monocotyledons. The algae. Algae are singular alga, members of a group of predominantly aquatic photosynthetic organisms of the kingdom Protista. Algae have many types of life cycle, and they are ranged in size from microscopic Micromonas species to giant kelps that reach 60 meters to 200 feet in length. Their photosynthetic pigments are more varied than those of the plants, and their cells have features not found among the plants and animals. In addition to their ecological rules of as oxygen producers and as the food base for almost all aquatic life. Algae are economically important as a source of crude oil, as a sources of food, and the number of the pharmaceutical and industrial products for humans. Algae are also defined as a eukaryotic. When we say eukaryotic, the nucleus bearing. So the algae are defined eukaryotic organisms that photosynthesize but lack of specialized multicellular reproductive structures of plants which always contain fertile gametes producing cells surrounded by sterile cells. Algae also lack true roots, stems, and leaves, feature they have with the vascular lower plants, or the mosses, liverworts, and hornworts. Now, what is the cyanobacteria? So, when we say cyanobacteria, it is the aquatic and photosynthetic that is live in the water and can manufacture their own food. Because they are bacteria, they are quite small and usually unicellular. Though they often grow in colonies large enough to see. Cyanobacteria are responsible for the Earth transitions from the carbon dioxide rich atmosphere to the present relatively oxygen-rich atmosphere as a consequence of oxygenic photosynthesis. Next is the Rhodophyta. Red algae, or the division Rhodophyta, any of about 6,000 species of predominant marine algae, often found attached to other shore plants. Their morphological range includes filamentous, branch, featured, and sheet-like thalli. The taxonomy of the group is continuous, and organizations of the division Rhodophyta may not accurately reflect the phylogeny or the evolutionary relationships of its members. Next is the Chlorophyta. The Chlorophyta is a taxonomic group of or a phylum comprised of a green algae that live in the marine habitats. Some of them are found in the fresh water on a land, while some species have even become adapted to thriving in extreme environments such as the desert, Arctic regions, and hypersaline habitats. Chlorophytes are a green algae, and as the name suggests, they are green in color due to the presence of chlorophyll present in the thylakoids. They also have other pigments such as the xanthophyll and the carotene. Well, depending from a species to a species, they can be unicellular or a multicellular. Next is the Heterocontyphyta. Heteroconts are a group of protists or are formally referred to as Heteroconta, Heterocontae, or Heterocontyphyta. The group is a major line in eukaryotes. Most are algae, ranging from the giant multicellular keep to the unicellular diatoms, which are the primary component of the plankton. Next is the Dinophyta. Dinophyta are divisions of an algae which are sometimes alternatively regarded as the protozoa or the class of the Phytomastiogophora. They are mainly 45 unicellular. The cell typically has a transverse furrow or the girdle and the longitudinal furrow or the sulcus. The girdle divides the cell into two halves called epicone and hypocone. Next is the Cryptophyta. The Cryptophytae are a class of an algae, most which have a plastids. They have about 220 species are also known, and they are common in fresh water and also occur in marine brackish habitats. Each cell is around 10 to 15 in size, meters in size, and flattened in each shape, with an anterior groove or pocket. At the edge of the pocket, there are typically two slightly unequal flagella. The Cryptophytes are distinguished by the presence of characteristic extrosome called ejectosomes or ejectosomes, which consist of two connected spiral ribones held under the tensions. If the cells are irritated either by mechanical or chemical or a light stress, they discharge, propelling the cell in the zigzag course away from the disturbance urge ejectosomes visible under the light of the microscope.